Hello, everyone. Welcome to the UPLB Museum of Natural History's Biodiversity Seminar Series. I am Michelle San Pascual, your webinar host for this afternoon. To those who are new guests here, we are glad that you have joined us. To our regular participants, we thank you for your continuing support of our webinars. Okay, I'm glad to introduce our speaker today, Ms. Marnelli S. Alviola. Marnelli is a University Research Associate at the Institute of Biological Sciences, College of Arts and Sciences, UPLB. She graduated with the degree of Master of Science in Forestry, Forest Biological Sciences in the University of the Philippines Los Baños last June 2019. Her master's thesis entitled Collembola Diversity in the canopy and forest floor of different vegetation types in Mount Makiling, Philippines, is the first comprehensive taxonomic study of Philippine springtails after almost 4 decades, and also the first in the country to document both the taxonomy and ecology of canopy Collembola. She is also project staff in the DOST Nicer Caves program and of the Philippine Terrestrial Arthropods Biodiversity Survey, or PhilTops. Everyone, let's all welcome Marnelli Alviola.
Thank you very much, Ms. Alejada, for the introduction. Good afternoon to and welcome to all of our participants. For this afternoon, we will be talking about springtails of Mt. Makiling. On behalf of my co-authors, Dr. Felipe Soto Adames, Mr. Christian Locanias, Dr. Virginia C. Cuevas, Professor Juancho B. Balitibat, and Dr. Irneo L. Lit Jr., I will be presenting our recently published paper in the Philippine Journal of Science entitled Collembola Assemblages in the Canopy and Forest Floor along an Elevational Gradient at Mt. Makiling, Philippines. Before we start with the introduction, and for those of you who are not familiar with springtails, I would like to show you first a very short video on how Collembola or springtails look like under a dissecting microscope. So here it is. These are two individuals from the family Hypogastruridae. So, what are springtails? First, they are the members of the class Collembola. And Collembola is a class under the phylum Arthropoda and subphylum Hexapoda. The other more popular member of the Hexapoda is the class Insecta. So, springtails are actually not insects. Next, they are small animals. Most are only a few millimeters long. And although some species of family Unicuridae can sometimes grow up to 9 millimeters. Next, their most obvious feature or popular feature is their jumping organ or furcula. This is their furcula, and others call it as springing organ as it enables them to spring rapidly into the air. This is also where they got their common name, which is springtails. Next, the majority of springtails feed on fungal hyphae or decaying plant materials. In the soil, they are known to influence the growth of mycorrhiza and control fungal diseases of plants, which are known to be generally beneficial. Next, they are generally short-lived. Few survive as adults for more than a year or two, although some cave species and those living on cold climates may live longer. What else? Well, there are around 6,500 species described worldwide. And they have a very wide global distribution and abundant on every continent, including Antarctica. When I say abundant, they are extremely abundant in soil and leaf litter, sometimes occurring at 10,000 to 100,000 individuals per square meter of soil. But because of their small size, their contribution to total soil animal biomass is low, but it should be noted that they are extremely important in influencing the structure of some soils. Unfortunately, even though they are known to be abundant and ecologically important, they are still regarded as one of the most neglected arthropod taxa in the Philippines. Here, I just want to give you a few information on their general morphology and classification. Springtails have two forms, the elongated form and the globular form. The elongated forms are further divided into two orders. We have order Poderomorpha and order Entomobryomorpha based on the distinctness of their first thoracic segment or the prothoracic segment. Order Poderomorpha have a distinct prothoracic segment, while Entomobryomorpha have an indistinct prothoracic segment. When you say prothoracic segment, it is the first segment of their thorax. On the other hand, the globular forms also have two orders. We have order Symphipleona and order Nilipleona, and they are divided on the basis of the length of their antenna relative to the length of their head. Order Symphipleona have antenna longer or as long as their head, while members of Nilipleona have antenna shorter than their head. On the other hand, the canopy science is also a relatively new field in the Philippines. The canopy has been considered as the last biotic frontier by Erwin in 1983 due to the favorable habitat it provides to numerous organisms that are rarely or never been found on the forest floor. As a matter of fact, explorations of the forest canopy have increased the global estimates of biodiversity by several folds. The canopy also serves as a dynamic interface between organic nature and the atmosphere, which is a great avenue in addition to the most studied elevational gradients for studying how organisms react with changes in the environment. This study is the first conducted on springtail diversity along an elevational gradient and also the first published report of canopy arthropods in the Philippines. This study was conducted to determine patterns of distribution and diversity of springtails along an elevational gradient in the canopy and forest floor of Mount Makiling, Philippines. We hypothesized that one, springtails would respond strongly to both elevation and strata, as these parameters possess a distinct set of environmental conditions that could have discernible effects on springtail assemblages. We also expect that a clear distinction between forest floor and canopy species exists. Our study site. The study was conducted in the northeastern side of Mount Makiling. Sample collection was conducted August to September 2018. So we selected 3 natural forests at different elevational sites. The first site, the lowland evergreen rainforest, is composed of early to late secondary growth forest at an elevation between 442 to 655 meters above sea level. The second site, the lower montane rainforest, spans from 760 to 899 meters above sea level, with vegetation consisting of natural stands of old growth forest with prominent vines and lianas. Human activities starting from this area, aside from hiking of mountaineers, are already limited. And then the last site, the upper montane rainforest, starting from 900 meters above sea level, is composed of trees which are significantly shorter and covered with thick layers of mosses. As expected, soil pH and temperature decreases with increasing elevation. Sample collection and processing. At each of the 3 sites, a 20-meter transect line was established based on rapid assessment of suspended soil presence and accessibility from the trail. 12 suspended soil samples and 12 forest floor samples were collected in each vegetation or elevational type. For the whole study, 36 canopy or suspended soils and 36 forest floor samples were collected representing the 3 vegetational types. We refer here to suspended soils as the soil and leaf litter present on tree holes, branches, epiphytes, among others. While the first branch of the tree up to its total height was considered part of the canopy. Next, arthropods were extracted for a period of 1 week from the soil samples using a burlesque tolerant funnel. And all springtails were sorted under a dissecting microscope. And then all springtails were separated for slide mounting, while the other arthropods were set aside and placed in microtubes for future studies. Lastly, the springtails were identified to the species or morphospecies with the use of several dichotomous keys and illustrations. Next, data analysis. The data gathered were subjected to several statistical analysis using paleontological statistics and estimate S. Rare fraction curves were used to check if sampling effort was enough to properly assess the diversity of the arthropod assemblage in the site. And then true species richness values were estimated using Chow-1 estimator. And then Simpson diversity and dominance indices as well as Pilus dominance index were computed. Indices of each forest type and strata were compared using Hutcheson's diversity t-test. And then unique and shared species were noted, and similarities between samples were analyzed using Bray-Curtis and Jacquard similarity indices. On the other hand, the effects of elevation and forest strata on species richness and assemblage were assessed using two-way permutational multivariate ANOVA or PERMANOVA, and then post-hoc pairwise comparisons were made, and then pairs with significant differences in PERMANOVA were subjected to similarity-dissimilarity analysis using SIMPER to determine which taxa contributed to the different species. I hope that it will be easier to understand when you see the results. So, results and discussion. A total of 2,287 springtails representing 31 morphospecies and 8 families were collected during the study. The canopy contributed 1,458 individuals from 27 morphospecies and 8 families of Collembola. On the other hand, the forest floor contributed 829 individuals from 20 morphospecies and 7 families of Collembola. Now, the table here summarizes the number of species and total abundance observed at each site per strata. As you can see, the lower montane canopy has the highest number of species, while the upper montane floor has the lowest. In terms of total abundance, the lowland canopy has the highest number of individuals, while again, the upper montane floor has the lowest. I just also included here the true species richness estimates from Chow-1 and from rarefaction curves. However, you can see that the completeness ratio for all sites was less than 85%, indicating that not all Collembola species from each assemblage are represented in the sample. Next, this figure shows the A, the mean species richness, and B, mean overall abundance of springtails in the canopy in pink and in the forest floor in gray. As you can see in A, mean species richness was consistently higher on the forest floor at all elevations, although the standard deviations of all sites indicate no significant differences in the number of species. On the other hand, in B, mean abundance did not show a consistent pattern and a significant difference was only observed in the lowland forest, in the lowland forest, unlike what this is actually unlike what we hypothesized, because only the mean abundance was found to be higher in the suspended soil than the forest floor. The higher surface areas in the forest floor compared to the canopy layers may have contributed to the higher mean species observed in the forest floor. However, it should be noted that in this study, as well as several other studies, it was observed that the suspended soils were actually more densely populated compared to the forest floor. Pauletti in 1991 noted that canopy soil contains a higher concentration of minerals and nutrients, highlighting that they really have the ability to cater more species. Next, the diversity indices showed an interesting pattern. As you can see, there is an increasing trend of diversity. As you can see, there is an increasing trend of diversity in the canopy with response to increasing elevation. This is in contrast with the forest floor species with decreasing diversity with response to increasing elevation. Consequently, the evenness index followed the same trend as that of diversity indices, while the dominance indices had an opposite trend. The pattern of diversity observed in the canopy springtails followed the same pattern of the edaphic arthropods in general, as reported by Sadaka and Ponge 2003, and also of Kutzpool et al. In 2010. On the other hand, the forest floor springtails concur with the diversity patterns observed in mammals by Haney in 2001, as well as with birds and vascular plants observed by O'Donnell and Kumar 2006, which decreases with increasing elevation. Our results suggest that springtails may follow the arboreality hypothesis in frogs, which is proposed by Sheffers et al. In 2013, in which arboreality increases with increasing elevation. These results were actually important in predicting how the organisms would respond to increasing temperature as a result of climate change. Aside from this, the prevailing environmental conditions might also have immense effects on the decreasing total abundance and richness from the lowland to the montane forest. With respect to elevation, temperature and soil pH also decrease from the lowland forest to the upper montane forest. The species of Collembola differ in their preferred habitat, temperature range, and temperature range, but the majority of species live in regions without extreme cold temperatures. Though the temperature in the lower and upper montane forest is not yet physiologically damaging for more springtail species, our results suggest that the temperature in the lowland forest is favored by most springtails. The decreasing abundance and richness with increasing soil acidity also suggests that springtails, just like most organisms, prefer basic than acidic soil. Collembola species have been documented to show sensitivity to changes in soil pH. They are also being used as bioindicators on changes in land use. So they really are very sensitive to soil pH. Next, species composition. As I've mentioned, a total of 31 morphospecies from 8 families were collected. 5 species were present on all samples, while 13 species were present in all elevations and 16 species were present in both strata. 3 species were unique in the lowland forest, 4 in the lower montane forest, but none in the upper montane forest. Meanwhile, 11 species were unique in the canopy and 4 from the forest floor. As you can see in this figure, each stratum in general and per elevational type also harbor unique species. So they all have unique species. So we cluster the sites based on the similarity and similarity of species. Next, you can see that the clustering analysis based on the assemblage from breaker to similarity index revealed 3 groups for the samples. So we have all the forest floor samples. We have the lower montane and upper montane canopy, and lastly, the lowland canopy. The same pattern was observed in B, the Jacquard Similarity Index, except that the lowland canopy is grouped closer with the forest floor samples. So what does this mean? So the distinct springtail communities along an elevational region may be linked to the known effects of elevation on the properties, temperature, and moisture of the soil. In addition to the effects of elevational gradient to the soil, the changes in the vegetation composition may also have contributed to the distinct communities per elevation. Faunal boundaries. Several studies have also found faunal boundaries between the soil or litter layers, between the canopy layers, and sometimes between the upper and lower canopy levels. This may be well pronounced in tropical rainforests like Mount Makiling with well-delineated canopy layers. Also, the food preferences of arthropod varies, in which fungivores and herbivores are most likely to prefer the canopy layers, while the food generalists, including scavengers and detritivores, usually forage on the forest floor. Springtails, being mostly detritivores and fungivores, may be expected in both strata. Species assemblage from the forest floor are closely related to one another. Unlike that of the canopy, where only the upper montane and lower montane are similar. The shared species between elevation and strata suggests that several species of springtails are able to move between the forest strata and forest types. Despite being wingless, the springtails are able to disperse over long distances. Bassett in 1998 noted that the occurrence of canopy-dwelling springtails, mites, as well as arachnids on the forest floor or montane forest may actually be attributed to the moss and epiphytes on a tree trunk linking the 2 strata. Despite that, the springtail assemblages between the canopy and forest floor of both montane forests in this study remains still significantly different. Next, we have the permanova. So the results of our permanova, as you can see here, indicated significant effects on elevation, significant effects of elevation on species richness and of strata on both species richness and assemblage. Both elevation and strata were also found to have significant effects on the overall assemblage of the springtails. The interaction of the elevation and strata was found to be insignificant in this analysis. Our post hoc analysis indicated significant differences in springtail assemblages in the following. Number 1, between strata in all forest types. 2, lowland floor versus upper montane floor. Lowland canopy and lower montane canopy. And lastly, lowland canopy and upper montane canopy. So based from this, we analyzed them using SIMPER to determine which taxa contributed to to these differences. So as you can see here, 6 morphospecies were found to highly contribute to the significant differences observed in the springtail assemblage. 4 of these species were from Lepidocyrtus sp4, sp2, Coeroliucinctus, and sp1, Lepidurtus, while the other 2 species were from Cryptopagus sensilato and Aloscopus. All of these species were observed in all study sites in both the canopy and the forest floor. Looking separately in the canopy and in the forest floor, it can be noted that the higher elevational sites did not differ significantly in springtail assemblages. The adjacent location of these study sites, the lower montane and upper montane canopy, in addition to almost the same prevailing environmental conditions, may have accounted for this result. In the canopy springtail assemblage, significant differences were only observed when being compared with the lowland forests. Also, in the forest floor assemblage, only the upper montane floor and lowland forests were found to be significantly different. In addition to Lepidocyrtus species, another species from Aloscopus has highly influenced our results. So these are the species which accounted for the significant differences we observed from different sites and strata. So for conclusion, the data analysis used in the study generated several significant results despite the fact that the completeness ratio of all sites was less than 85%. The faunal composition of springtail assemblages showed a significant response in both elevation and strata, canopy versus forest floor, in Mount Makiling, Philippines. The forest floor species assemblage are closely related to each other, unlike those of the canopy, where only the upper montane and lower montane are similar. The distinctiveness of these springtail assemblages in the canopy and higher elevational areas suggest a need to improve our biological data in areas or habitats often neglected by standard biological surveys. Thank you very much. I would like to acknowledge the DOST Picard, DOST Science Educational Institute, NRCP, and Center for Systematic Entomology for funding this research. I also would like to thank the following institutions: Institute of Biological Sciences, UPLB Museum of Natural History, Florida State Collection of Arthropods, and the Makiling Center for Mountain Ecosystems. This study is also part of our initiative under the Philippine Terrestrial Arthropod Biodiversity Survey, or PhilTabs. Thank you very much.
Thank you very much for your interesting presentation, Ms. Alviola. And for the Q&A portion, Dr. Jennifer Niem will be our moderator. She is one of the university researchers of the University of the Philippines Los Baños Museum of Natural History and also the curator of the Mycological Herbarium. Dr. Niem, kindly join us.
Thank you very much, Michelle. If you have any questions, so just type them in the chat box and I will read them for you. But to our audience who might want to ask live questions, you may also do so. Just unmute your microphone and open your video. So our open forum is now officially open. And let me check the chat box. So there are no questions yet. So let me throw in the first question. So, Nelly, can springtails be classified as insects, although they do not have wings?
No, yes, they are wingless. Maybe I forgot to mention that, but that is actually not the reason why they are not considered as insects, because historically they were classified as insecta under the Arthropoda, but recent molecular evidences as well as their mouth parts. That's actually the main reason. Their mouth parts is entognatus or entonatus, meaning it is retracted from the head, which is in contrast with all of our insects, which have ectonatus mouth parts or external mouth parts. So yun. I hope I answered your
So,
question.
hindi siya insect.
No, they are not
Okay.
insects.
So, in connection with the springtails being wingless, you mentioned that you found in your research, you found a high number of species in the canopy. And I would think that this is strange because being wingless, you wouldn't expect na mataas yung population niya sa canopy. So, could you think of reason why um, you found high number of Collembola species in the canopy? I know you mentioned earlier yung mosses sa bark na this is a possible contributing factor, but meron pa kaya na ibang reason?
Yes, it is actually very interesting to see wingless arthropods in the canopy, sometimes up to 30 meters. Um, in terms of springtails, as I have mentioned a while ago, the mosses in the bark, which may provide continuity to from the forest floor to the canopy. And aside from that, I think I have mentioned a while ago that they have the furcula, which allows them to spring or to jump from one area to another. In addition with furcula, they also have this ventral tube, which is common to all springtails. And this ventral tube functions for adhering to surfaces. So and in addition to furcula, the ventral tube may help the springtails to migrate vertically. Aside from that, they are very tiny. So they are very tiny and lightweight. So it is easier for them to be carried by the wind from the floor to the canopy. And what else? Maybe foracy is also possible, is that they may attach or ride from other flying insects so that they can be transported to to higher areas in the canopy. Yun
So,
lang
so
po. Yun
pero
yung
so
naisip ko.
it's really common to find sa
Actually,
canopy yung mga springtails.
marami po. Actually, even in helicopters, they were using net along in helicopters, and they actually see several springtails. Yeah, I think it's because they are lightweight. Most of it is because they are lightweight and can be easily carried by the wind.
Do we have questions from the audience? Anyone who wants to throw in a live question, please do so if you want, or just type in the chat box if you have, um, if you're shy to ask a live question. So, so another question. Um, you mentioned earlier about springtails feeding on fungal hyphae. Um, Meron bang specificity ang feeding pattern ng springtails, like preference of a certain group of Collembola species to a particular fungal species?
Yeah. Well, actually, Collembola graze from a wide variety of sources. There are Collembolas which are generalist in terms of general fungivores, but there are also several species which were found to be selective in grazing. So I can think of Sinella. Sinella is from Entomobride, which its feeding actually suppressed the growth of Fusarium oxysporum, which is, I think it's it was wilting from cucumbers yata po. So that's one example of springtails being selective or specific in certain type of fungi. But pwede rin po siya maging harmful because there is also a species of Hypogasturidae which are considered as pests of commercial mushrooms. So yes, there are several species which feed specifically on one group of fungi.
So yung sa mushroom, kinakain yung mismong fruiting body?
Mm-mm. And they sometimes occur in very high numbers, as I've mentioned. Minsan may swarming. So their feeding habit may really cause economic damage.
Yung sa mga commercial mushroom?
But the positive side is there are several examples, hindi lang ako makaisip ng specific at this moment, that they are now actually being considered as a biological control agent of fungal diseases in plants, na if it is possible that na ilagay talaga sila sa soil to suppress the growth of the fungi. So marami pong ganong studies ngayon na ginagawa.
Okay, thank you. Finally, we have a question from the audience, and it's from Dr. Factory Regent Emily Dupo. So, do you think soil pH, temperature, and texture influence collembolan survival and distribution? And another is, can these animals serve as good subjects for science investigatory projects? Because we have teachers and students watching in our Zoom right now.
In terms of the environmental factors such as soil pH, temperature, and texture, I think this is a very good topic. We actually did include these environmental factors in my master's thesis, but due to several factors, I think it's best to do it again because of that time we do not have enough equipment to do this. But since springtails are very sensitive to soil pH and temperature, I think this will be a good subject. And as I've mentioned a while ago, they are commonly used as bioindicators. So pwede po, maganda po siya. It's a good topic. And in terms of can this animal serve as a good subject for science investigatory project? Yes, I think though we have perhaps difficulty in identifying them into species level or even genus level because they should be mounted first before we are able to identify them, and it will be difficult for other schools which do not have actually the enough facilities for that. Pero
Pwede naman hindi siguro taxonomic work, kuwari effect lang ng temperature sa
pwede
isa-subject
also,
ko yung Collembola sa different exposed sa different temperature
or
na ganun,
yes,
no?
kung abundant
Pwede
yung i-consider, yeah, that will be a
pwedeng
good
mga investigatory
subject.
project pang high school. Parang iba-ibang temperature gradient lang, no? Yeah. Any more questions from our entomologists in the audience? We have another question again from Mark June Alcantara. So he said, there's a comment, thank you for the presentation and for documenting Philippine canopy through arthropod fauna. He has several questions. The first one is, environmental conditions in canopy and understory of tropical forests are very distinct and could vary along the vertical gradient. Do you think the variable vertical height could have contributed to relatively high diversity in the canopy?
In the first question, yes, there are actually several studies, as I've mentioned, that there sometimes exists a faunal boundaries even within the vertical strata, from the forest floor to understory and to the canopy. I think there might also be differences, but for this study, we just actually considered the canopy, starting from the first branch of the tree up to the height of the tree as the canopy. So perhaps in the next studies you can do the vertical stratification, and let's see if we can observe significant differences.
Okay, thank you. Second question, any potential effects of different suspended soil volumes or area effects?
Actually, I didn't mention that. We actually, initially, ideally, our soil sample should be around 1 kilogram, but it was a great challenge to collect 1 kilogram of suspended soil in the canopy, especially for certain vegetations na konti lang po yung suspended soil. So, yes, that there may be some effects with that. That's why I mentioned a while ago that they may be densely populated, but we did not consider the volume in this study. That's why we also base our explanations based from the mean abundances and mean species richness. Next.
So another question is, would it be possible to partition the effects of elevation and vegetation type with your data set?
Yes, actually we did that. Perhaps you could our publication is I think it's free access no? At the Philippine Journal of Science website.
So separate yung effect ng elevation and vegetation, the interaction. Separate yung
for
analysis.
complete discussion.
And Sir Jun would like to ask a live question. Please, Sir Jun. Sir Jun. Sir Jun here. Sir Jun. So we have another, we'll ask Sir Jun later. There's another question from Faye Daniel Dacosta. Good afternoon. Where can we see the pictures of the species that were found? And where can we learn more about Philippine Collembola? It's in your publication, I suppose?
Actually our publication is pictures. No.
Walang pictures.
Yeah, it's more on the data and a list of the species. But if you would like to see pictures, perhaps you could visit UPLB library. My master's thesis include pictures of all springtails collected from
Or
this.
perhaps if you're really interested, maybe you can send an email to Nelly, and then Nelly will send you some pictures. We will type in Nelly's email address, if that's okay, Nelly.
Yes,
Later.
of course.
Yeah. Okay, Sir Jun.
Where can we learn more about Philippine Collembola? As I've said, it is we have few literature on Philippine Collembola, and we're just starting to study. So there isn't several literature available in Philippine Collembola at this moment. But if you are interested with the world Collembola, we have this checklist of Collembola of the world available online. You can just type it in the Google. So yun po.
Sir Jun, would you like to answer?
Hello.
Ask your question?
Pwede
yes.
na?
Opo.
Okay. Actually, this is not a question, but a comment. I'm actually very happy and very proud that Nelly started studying Collembola. Maybe just to emphasize, the last work before Nelly's was Dr. Gapud's series in the 1970s, and therefore really this is something that we should that updated our knowledge, and that's why related din dun sa question ni Dr. Nyem kanina, we have been traditionally educated or taught that Collembolans and other entonatus arthropods are insects. Well, in older textbooks, they still are classified under insects because they have 6 legs. But as Nelly has expounded, they are technically now a separate class and order. And then also traditionally, we have been taught that if you want to see Collembola, look at the soil. So it's good that Nelly has shown through her thesis, our paper, that mas marami pala dun sa taas sa canopy, and they're actually contributing to the ecology of the soil in the canopy, and therefore also to the nutrient recycling doon sa canopy mismo. So for the question about by other students na paano mo mag-aral ng Collembola, maybe this is already time na at least nandiyan na yung nag-start. I'm sure Nelly will continue studying Collembolans and other microarthropods in the soil, and so maybe she can be your mentor. So thank you very much.
Thank you, Sir Jun. So, ang tagal na pala nung last study prior kay Nelay. Good job, Nelay. So, we have another question from Nieves Capili. Can the presence of Collembolas be correlated to levels of floral diversity?
Yeah, it's possible. We were actually planning to do that, but we did not conduct a vegetation analysis during my thesis. But we think that it is really an interesting topic to study in the future. Yeah, you can also do that if you really are interested. Pwede po yan. I think there is a significant difference na makukuha po nila.
So, any more questions? Okay, I'm just wondering, meron ka bang mga na-encounter na challenges, particularly in your sample collection? Kasi as you mentioned, this is the first studies in canopy arthropods sa Pilipinas. Tapos sabi nga ni Sir Jun, yung published, oo,
Yeah.
published. Tapos yung sinabi nga ni Sir Jun, matagal na yung nauna pa na nag-study. So, sa sample collection ba nagkaroon ka ng mga problema sa...
Yes, it is actually very challenging to... The first challenge that we encountered is how to collect arthropods from the canopy. Because based from most of the literatures that we see are using insecticide fogging or canopy fogging. So they use insecticides. And they really get sometimes millions of arthropods. But we can't do that here, especially I don't think MACME will grant us permit if we will conduct fogging. And we think that it is too invasive. And I think it is important that I know that my target organisms are usually in the soil and leaf litter. So I decided to just focus on one collection method, which is suspended soil collection. But there are still challenges because we have to get a tree hiker, professional tree hiker, and make sure that they are using safety equipment for safety.
Yung may harness.
No po. And life insurance. Buwis buhay po.
Okay, so have you got any more questions?
And I just want to share that we are working on a core-funded project, which is a canopy project of Mount Makiling. So we are trying to test if those methods, collection methods that we usually use on the floor can also be used in the canopy. So it's just starting. So hopefully we will be able to share the results with you soon so that it will help other researchers on how to collect arthropods from the canopy.
I don't see any more questions in the chat box. So I guess if we haven't got any more questions from the audience, I will now turn you over to our MC. So Michelle, take it away, please.
We would like to give this certificate of appreciation to Marnelli S. Alviola, for serving as resource person during the UPLB Museum of Natural History Biodiversity Seminar on Springtails of Mt. Makiling, Collembola assemblages in the canopy and forest floor along an elevation gradient at Mount Makiling, Philippines, held today, July 6, 2022, at 2:30 to 4 p.m. via Zoom, signed by our director, Dr. Marian P. De Leon. Okay. So if you want to know more about the Museum of Natural History, you can visit our website. That's https /mnh.uplb.edu.ph. You can also email us at mnh.uplb@up.edu.ph. For those who haven't like or follow or subscribe, you can do that after this webinar. So please visit our Facebook, Twitter, YouTube, and Instagram. So that's UPLB Museum. You can also check us out at Wikipedia and TripAdvisor.